The Food and Drug Administration, or FDA, is the governmental body that regulates the safety of additives used in foods grown in the United States. The FDA defines genetically modified foods as food products produced through modern methods of biotechnology, such as recombinant DNA techniques and cell fusion. According to the FDA, the European Commission refers to these foods as genetically modified organisms. The United States uses the term genetic modification to refer to all forms of breeding, both modern, i.e., genetic engineering, and conventional.
Genetic modification of crops and domesticated animals has been an integral part of agricultural practices since human beings first began cultivating their food. However, conventional practices of breeding and selective crossing have been replaced by modern biotechnologies that allow for the transfer of genetic material between organisms. This process is unprecedented. It is the first time that human beings have directly altered the structure of genetic material of organisms intended for human consumption. In 2004, 85% of all soybeans, 75% of all cotton, and 45% of all maize grown in the United States were genetically modified in some way.
How are foods genetically modified? Plants are genetically modified mainly by 3 different techniques, all of which result in a plant with altered genomic material and consequently with a food complete with sought-after characteristics. The techniques are agrobacterium insertion, electroporation, and particle bombardment.
The basic technique of agrobacterium insertion is to use a tumor-inducing bacteria to insert desired DNA sequences into the target plant. First, the desired sequence of DNA is placed into a plasmid ring. This ring is then placed into the Agrobacterium tumefaciens. Once the Agrobacterium tumefaciens is introduced into the host plant, it transfers the new genetic material into the host's genome using the mechanisms that it would normally use to cause tumors. The plant is now said to be transgenic, which means it contains foreign genes, and will transmit these genes to its offspring, creating a new generation of plants with the desired features. Maize, rice, barley, and wheat are all easily transformed by this technique.
The second method of gene transfer is electroporation. In this method, high-voltage electrical pulses are shot at plant tissues that contain its DNA. The pulses cause pores in the plasma membrane to open. This allows the recombinant plasmids to enter the protoplasts and change the genetic material in the nuclei of the cells.
Particle bombardment is the third major technique used to alter the genome in plants in agribusiness. Gold or tungsten beads coated with RNA or DNA are shot at high speeds into plant cells by a gene gun, delivering new messages into the nuclei of the plants to be transcribed and passed on to future generations.
What foods are likely to be genetically modified and for what reasons? Fruits and vegetables of all kinds are altered for many reasons. The tomato is an example of an organism whose genome is altered by farmers so that the fruit will be redder and thus more attractive to buyers. Delayed ripening is also selected for because an unripened tomato is less likely to be bruised or damaged during travel. The tomatoes are then sprayed with ethylene to promote ripening once they have reached the supermarket.
Genetic engineering is not only used to improve the beauty of food, it is also used to improve its quality. Modifications to potatoes include increasing starch content. A potato with this modification absorbs less oil when fried than would an unaltered potato. Therefore, in some cases, genetic modification can lead to a healthier product.
Soybeans and canola have both had alterations made in their fatty acid pathways that reduce the amount of saturated fat found in the mature plant.
Vitamin A deficiency is a major problem in many Asian countries, where rice makes up a significant portion of the diet. Beta-carotene has recently been introduced into a species of rice in an effort to combat this form of malnutrition. Beta-carotene was chosen because humans convert it to vitamin A during digestion. Because rice is so cheap to produce and so widely consumed, this development will significantly reduce the problem of vitamin A deficiency in Asian countries that suffer from lack of vitamin A availability. In conclusion, one of the pros of genetic modification is that it increases crop yield, which makes it possible to feed more people using the same quantity of land under cultivation. Advocates of genetically modified foods cite their relatively low production costs as a plausible solution for world hunger. As seen with beta-carotene-induced rice, genetically modified foods may also be used to reduce malnutrition.
However, it is unclear what the interaction will be between genetically modified food and their uncultivated surroundings when these crops escape from farmers' fields. As with any new developments implemented on such a large scale, the side effects of genetic modification, including unknown health effects and unforeseen ecological impacts, warrant close observation and further investigation.